Hello students, welcome to EPG Pathshala. I am Dr. Shashi Kumar from International Center for Genetic Engineering and Biotechnology. Today we are going to discuss about module Chloroplast Transformation 1 under plant biotechnology and crop improvement. The learning objective for this module of chloroplast transformation, we will discuss how many types of chloroplast, what is the method of chloroplast transformation, how you confirm the chloroplast transformation, and then comparison of chloroplast transformation with the nuclear. We do like chloroplast and nuclear transformation, and then application of chloroplast transformation. Chloroplast transformation introduction. Chloroplast genome resemble to cyanobacteria. In past, there is a possibility that eukaryotic cells has got infection of cyanobacteria and then they have the symbiosis for long time, and that might have given rise to the algae and then further plants. So these are the mixture of cyanobacteria and eukaryotic cells has given rise to the modern plants. Chloroplast genes are regulated by both NAP and PAP promoter. NAP is nuclear encoded promoter, PAP is plastid encoded promoter. So if your gene present into the chloroplast, then outside is the nuclear genome which controlling the genes expression into the chloroplast. So the beauty of the chloroplast that they are controlled by both chloroplast itself and outside nuclear genome present also control the expression of genes. The innermost matrix in chloroplast is called stroma, which contain multiple copies of the chloroplast genomes. If you have one single cell, then there is a possibility that in one cell there may be like 5 to 10 organelle or 100 organelle present in one cell, and one organelle has about 100 copies of the chloroplast genome. So you can express lots of protein in one cell of the plant compared to the nuclear genome. Chloroplast transformation stages. There are different stages of chloroplast. Chloroplast can be present like in tomato in form of leucoplast, in rice etioplast, or in like in very primitive stage it can call as proplastid, and amyloplast in different form of food. So there is a different stages, but the particular things here to be noted that all the chloroplast is they have different shapes, different colors. The chloroplast genome is the same. There will be same nucleotide depending on what is the colors. So there are different stages. Like when we want to transform leaves, we use like in tobacco, tomato, potato, brassica, Rhodopsis and lettuce. We use leaf for the chloroplast transformation. So it's called chloroplast. But when we want to use transform like cotton, carrot, soybean, which are represented for regeneration via the like callus culture or somatic embryogenesis, they are non-greens, so we called like color plastids. This can be in different forms. We use like callus transformation for this. So generally like if you see the gold particle which is bombarded to the chloroplast organelle is bigger than the genome size of the chloroplast. So how the genome and this chloroplast vector is integrating into the genome of chloroplast. So this is integrating by the homologous recombination, which we will discuss later. So how the plastid and chloroplast transformation take place? Like in the first panel on the left hand side we have shown there's a chloroplast vector which we caught over the gold particle, and then it is propelled with the force by using the helium gas. It is propelled with a high speed and punctured through the leaf tissues and integrate randomly into the chloroplast. Then using antibiotic selection marker, the next panel you see that on antibiotics here we use kanamycin all the non-transgenic callus has died, but the transgenic callus is growing green at one corner. So this callus is further subculture on the new medium containing the antibiotics. That's how we can get the like transgenic chloroplast. Then we can transfer this into the greenhouse. Chloroplast transmission vector. Chloroplast transmission vector has the unique feature, like if you see that at the bottom, the vector has the left flank and the right flank. These left and right flank guide the integration of the above cassette into the chloroplast genome. So it is a typically like a very standard transformation vector, where you can insert there's a site in between two left and right flank. You can insert your vector, and using different promoter and the gene of interest, you can insert your genes within these two flanks, and then these flanks help integration of these whole cassette into the chloroplast genome. So left and right flanks are the one which guide the integration of your genes into the chloroplast genomes by homologous recombination. Plastid transformation. We have shown in more details how the chloroplast transformation or plastid transformation take place. You see that this is the typical chloroplast genome of carrot, which is Dacus carota. Here you have seen that there is a two black thick line is shown, inverted repeat A and inverted repeat B. So, for example, we take the native DNA of inverted repeat A and divide it like a small portion in like flank A and flank B, or left flank or right flank, and then put our cassette between these two native chloroplast DNA, and then by homologous recombination we help to get integrate this chloroplast vector into the chloroplast genome. So if you see that right, like inverted repeat A and inverted repeat B, they are the mirror copy each other. So if the integration of chloroplast vector at one side, at A or B side, then automatically in presence of Rec A enzyme, it will get automatically duplicated other side, because everything what is at the A side, it should be at the B side. So this is the beauty of the chloroplast transformation, that you inserting your chloroplast genome at one side, like at inverted repeat A, so it will also get integrated at other site also by the genes Rec A. This is like how you can transform the chloroplast we have discussed. So, but after the next stage, how you will confirm the chloroplast transformation has taken place or not? What is the status of what is the level, polarity level? Is it your chloroplast transformation is homologous or it's a heterologous? So this is very important to note, that if like it has been, you have achieved in chloroplast transformation homoplasmy or heteroplasmy. Like for example, in the first panel A, we can see left hand side panel A. This is the PCR, simple PCR. We can confirm by landing our primer on the gene of interest. We can find out on the gel that your gene has been integrated. Then there's the panel B. Again, there's for another gene you can cross confirm that this gene has been integrated. But how you will confirm that whether the genes which has been integrated is got the homoplasmy, it has fully mixed or homogenized with the chloroplast genome, or it is like there's many chloroplast genomes in one organelle, only like 10% is transformed or 50% transformed. So this we can find through the southern blotting. So panel C, you see that the first line, panel C, the line number 1 is the native, is just chloroplast genome, which showing the wild type chloroplast genome. But in second row, you see the two bands. The upper band is not much thick, so it shows that there are more chloroplast genomes which are wild type and less transgenic chloroplast genome in the plant number 2 or line number 2. In 3/4, you see the level of homoplast is increasing, and lane number 7, 8, that So that there is no wild type of chloroplast genome left. Everything is the transgenics. So this confirms the chloroplast transgenics. You can say transplastomic plants are 7 and 8. So this is the one way. Then this is we have done shown previously in the for carrot, and you can see that then you can do another analysis. But this is the basic methods how you will confirm the homoplasmy or heteroplasmy of genes which you have integrated in the chloroplast genomes. Chloroplast transformation advantage. The big advantage of chloroplast transformation that genes do not escape from one plant to another like in nuclear genomes. If in pollens, the mature pollens, the nuclear genome is present, but in chloroplast, in case of chloroplast transformation, there is no mature chloroplast organelle or gene present into the mature pollens. So when there is a breeding taking place between two different plants, so there is no like chloroplast genes is getting escape from one plant to another. All the chloroplast genes pass from one generation to another through seeds only. They cannot cross contaminate in the field. So this is the big advantage with the chloroplast, that the gene is contained. It does not help to produce the super weed as in case of the nuclear transformation. Chloroplast versus nuclear genome transformation. So we can do a chloroplast transformation by different way. First is like if you want to do the chloroplast transformation directly, then we can use the gene gun or electroporation of the isolated cells like you can say protoplast, which can also by you can use a PAG or different method to insert your genes. But generally in chloroplast, most popular method is the gene gun, which propel your DNA coated on the gold particle, and using the helium gas, we propel this DNA into the callus or leaves. So this is the direct method. The second method we can like use very well established technology, the nuclear transformation, like agrobacterium methods. In this is the mediocre method. The expression is not great. Here what we do the nuclear transformation, but the target the protein expression into the chloroplast by using the chloroplast transit peptide. Chloroplast and nuclear genome transformation, you will be interested that what is the difference between chloroplast and nuclear transformation, because both are the transformation. You putting the gene in chloroplast, some people putting the gene in the nuclear transformation. There's a unique feature of chloroplast and nuclear transformation. Like if you say a transgene copy, there's like more than 100 chloroplast genome per cell. So you inserting like more than 100 copies of your gene of interest. So expression is very high compared to nuclear transformation, where you can express 1 or 2 copy. Transfer frequency is low in chloroplast transformation, is challenging, difficult, but in nuclear transformation, the technology is well established, and worldwide every lab is who work in transformation is using this nuclear transformation methodology. Further, position effect not reported in chloroplast, but is reported position effect that where your gene is integrated, it can interrupt your important genes. So it is reported in nuclear transformation. In chloroplast transformation, everything taking place by homologous recombination. So you know where the gene is integrating and what is the site of integration. So there is no position impact here. Expression like of genes, like there is a polycistronic and monocistronic method in nuclear and polycistronic in chloroplast. Like if you say you can express multiple genes under one promoter, and for all these genes under one promoter there will one single mRNA. But in monocistric, that you need like one promoter, one gene. So if you have to express multiple genes, you need multiple promoter and multiple genes. So that's called like one gene and one mRNA. So there's a big difference that chloroplast express like multiple genes under one promoter, like long mRNA strands. Transgene escape and containment. The chloroplast has the beauty that the gene cannot escape through the pollens. While in the nuclear genome, the pollens is present, nuclear DNA is present in the pollen, so it easily cross-contaminate the other crops. While the chloroplast doesn't present into the mature pollen, so it will not spread to other crops. Toxic protein and accumulation within chloroplast, we have observed that the chloroplast may minimize the adverse impact of toxic protein, while the accumulation of toxic protein within the cytosol might lead to the serious pleiotropic effect. So it's not good to express this harmful protein into the nuclear genome. Functionality of proteins is slightly different. In nuclear genome, proteins are targeted to the endoplasmic reticulum for disulfide bond formation, while in chloroplast, disulfide bonds forms and protein properly fold, and it's fully active. So there's a different approach. And chloroplast can be the good mode to express the pharmaceutical proteins which don't need the glycosylation, and you don't need codon modification to express the gene. While in nuclear genome, like if you want to express Bt genes, you have to do the codon optimization to express the genes. Applicability chloroplast transformation have been reported for very few plants, while the nuclear transformation has been shown for a large number of plant species, though the level of protein expressed is low. Chloroplast transformation applications. Chloroplast transformation can be used for different applications like synthetic biology and metabolic engineering of isoprene pathway, that is isopentyl pyrophosphate of chloroplast for drug biosynthesis, or more than drugs, you can produce the different metabolites using the chloroplast transformation, using the novel new tools of synthetic biology and metabolic engineering. You can protect the crop using RNA and RNA like long standard expression in the chloroplast. You can protect the crops fully using this method. Cotton may be the one example which is highly infected by insect and pest. You can control all these insect pests using the long standard expression in the chloroplast. Chloroplast doesn't have RNA machinery, so it does not process the RNA into the chloroplast. So whatever the insect biting will be immediately killed when they are taking into the chloroplast transgenic materials. The further crop protection by expressing insecticidal genes, like in case of Bt, we have seen that when you express the Bt gene to the chloroplast without any codon modification, the impact on the insect is very high comparative to nuclear transformation. You can express in chloroplasts like prokaryotic or eukaryotic gene without any codon modification, which we will discuss further here. You can express lots of therapeutic proteins which does not need any glycosylation into chloroplast, and it could be tools to produce the cheap drugs by expressing into the chloroplast. So there's so many applications of the chloroplast transformation. One application we can say that to enhance the IPP biosynthesis, which is known to biosynthesize more than 50,000 molecules. In plant, there is a two pathway to produce the IPP, which is called isopentyl pyrophosphate. Like in a cytosol, there is a malonate pathway which produces the IPP, and in chloroplast, there is a MAP pathway which produces the IPP. So there is a two pathway which produce the IPP. So there may be interesting if we can increase the IPP, and then we can convert this higher IPP to produce some pharmaceuticals, or we can utilize it for some other application. Chloroplast transformation enhance IPP biosynthesis. So what we have done, like we know there's a two pathway producing IPP, one in chloroplast and one in cytosol. So what we have done, we wanted to increase the IPP level to see if we can use this system for producing some good molecules, some drugs. So what we have done, we have overexpressed in chloroplast the another pathway taken from the yeast and express the whole pathway into the chloroplast, and to see that we can increase the IPP or not. Interestingly, what we have seen that we were able to increase the IPP. Not only increase the IPP, we have shown here the functionality that yeast malate pathway is fully functional in the chloroplast and producing the IPP. Functionality was tested by, we put in a medium phosphoromycin, which specifically stop the expression of plastid map pathway. So IPP was not available to the plants. If you see the right side, the wild type, because the pathway which need for like photosynthesis, chlorophyll biosynthesis has been blocked totally. But the transgenic one, because they containing the IPP pathway from the yeast, so the end product IPP is produced, which is needed for the chlorophyll synthesis and the photosynthesis. So plants left side are healthy. So this way we have shown the functionality of the whole pathway in the using the chloroplast. So instead of two pathway, now these transgenics have three pathway which producing the IPP, which can be like further utilized for some useful application. You want to go in more details, you can go and study this paper, that how the whole process was carried out and how the remodeling of tobacco chloroplast was carried out to produce the higher IPP. Since we have shown previously that we can increase the IPP, which is required for different molecules, different drugs, biosynthesis, so what we have done, we have further extended this pathway and we diverted this higher IPP to produce like antimalarial drug Artemisinin. So the red genes showing that inside the chloroplast in a box dotted lines showing the yeast malate pathway producing higher IPP. So this IPP was further derived to produce the drug, and we put this Artemisinin biosynthesis pathway outside in like in nuclear genome and targeted to either mitochondria, like to produce higher amorphidine, or targeted to chloroplast to avoid the glycosylation process. So this way, smart way, we have produced the artemisinin, and you can see that in the right hand side that once you have dihydroartemisinic acid in presence of sunlight and oxygen, it automatically convert into artemisinin, and the plate showing the double transgenics nuclear and chloroplast, which have the like yeast and the artemisinin biosynthesis pathway. This is the one way how you can extend your pathway and produce the drug. Once you have the like some product you have produced using the chloroplast, then you have to show the functionality. Here we have shown the functionality of the artemisinin drug produced in plant. You see that left hand side there is a silent ring stage of malaria, plasmodium, which is quite infecting. So when we use the extract, our double transgenics plant, like artemisinin drug, it reduced this silent stage ring, reduced like a dot. So plasmodium is not able to survive because of this plant extract. So this way we showed the drug we have produced in plant is functional, and the further this confirmation was done into like by oral feeding to the mice also. We have shown that this artemisinin produced in transgenic plants is more effective than the pure artemisinin. The blue line show the efficacy of the drugs produced in our transgenics. So it's a nice example how we can use the chloroplast transformation technology to produce even drugs. Chloroplast transformation, one of the good example we have shown here, that how you can divert the higher IPP you produce in the plant for making it to drugs. If you want to go in more details, you should read this paper we published, like how we can compartmentalize metabolic engineering of artemisinin, which is anti-malaria drug, and we have shown the effective malaria treatment by oral delivery of plant cells. So for reference, one should read this paper for the applications and how diversion of higher IPP of chloroplast can be used to produce the drugs. So there's another application of chloroplast transformation like crop protection via long stranded RNA transform in chloroplast. You can see like in left hand and right hand side. Left hand side we see that is a chloroplast transformation, and right hand side show the nuclear transformation. When you express long RNA standard in nuclear, then there is a dicer which chop these RNA into small pieces. So when these bugs biting the leaves, they can compromise the impact of this siRNA. Whereas in chloroplast transformation, since there is no RNA machinery present in the chloroplast, so the insect when biting the leaves, they're taking in the long stranded RNA without any dicer chop kind of thing. So it's like double stranded RNA they choke taking in their body, and body has the dicer inside, and then it is chopped into small RNA you call small iRNA. So their impact is very lethal, and the insect immediately die. And so you can say the chloroplast transformation using long stranded RNA is more effective in controlling insect pest problems rather than the nuclear transmission. So this may be a great boon in future to controlling and to protecting our crops. It is another example how like insect resistant, salt tolerant, metal nutrients via chloroplast in hard plants species taking place. Like in soybean, you can develop the plant resistance by expressing directly gene to the soybean. In carrot, we have developed the high salt resistant, like up to 400 millimolar sodium chloride can be tolerated by this chloroplast transgenic carrot. Similarly, in cotton, we have shown that gene is not transferred to the other crops when you express the gene into the cotton chloroplast. So these are the nice example of containing the genes and the utility of these genes in the chloroplast transformation. It is another example if you want to express the genes without any codon modification, then chloroplast transformation is the best mode. Here you can do the transformation without any codon modification. Like for example, we express these amoebiasis lac-A gene to the chloroplast, which express up to total soluble protein up to 6% into the chloroplast. When we try to express this 65% AT-rich gene to the nuclear genome, we failed, we could not express. So this is the beauty of the chloroplast transformation, that it can express both eukaryotic and prokaryotic genes without any codon optimizer. Here we have shown that if you can express the protein in bulk amount using the chloroplast transformation, then there's a possibility that we can reduce the extraction and purification cost. We can simply the plant which expressing the protein, pharmaceutical protein in chloroplast. So we can simply take out these leaves and grind and dry and put in a form of capsule and deliver the drugs to treat the human being different disease. This way the extraction and purification costs, which make the drugs very costly and poor people cannot afford, that cost can be cut by using in Ayurveda system. We have used this grinding the tissues and giving as a cox as a drug. Same way we can produce the drug into the edible plant, grind them and put in a major drug into the capsule and deliver to the human being to avoid all the purification and extraction cost. This way the drugs have been tested. They are durable more than a years, and that way is the best way. I think in future the direct chloroplast transformation and then putting the drug in form of capsule could be the one another strategy to deliver the drug for treatment of the human beings. So once you did the chloroplast transformation, there is a important that you need to take out the antibiotic selection marker, which may not be needed anymore. So here we have shown the strategy how precise excision of plastid DNA can be taken place by these recombinase enzymes. So this is the methodology we have reported, which students can go in details for further if they are interested to know how to remove the unwanted gene from the chloroplast. Conclusions. First chloroplast transmission was reported in Chlamydomonas renhardi, which is a green algae. In most of the report, gene gun or ballistic method has been used for the chloroplast transmission. In chloroplast transformation, the gene integration takes place via homologous recombination. Chloroplast transmission is mostly reported in tobacco, uh, few reports in like other higher plant species like cotton, carrot, mustard, soybean, but there is very few reports in other species like wheat kind of things. Higher level of foreign protein expression due to multiple chloroplast genome per copy has been demonstrated using chloroplast transformation. Chloroplast transformation is very useful for generating like herbicide resistance, insect resistance, fungal resistance, abiotic and biotic resistance, and particularly biosynthesis of pharmaceuticals and metabolic drugs using new approach of metabolic engineering. Thank you.